Okay, so welcome to this video. In this video what we're going to discuss is the SREBP pathway, okay, which is a pathway for regulating the absorption of cholesterol molecules by cells and also the production of cholesterol molecules by cells, but through the level of cholesterol that the cell currently has within its cytoplasm. Okay, so this is the SREBP pathway, okay, and SREBP stands for the sterile regulatory element binding protein pathway, and we will see the sterile regulatory element binding proteins later on. Okay, right. So before we actually go into the details of the SREBP pathway, what I want to firstly do is discuss the ways that cells can top up the level of cholesterol that they have within their cytoplasm, okay, because there are two main ways. Either they can synthesize it de novo, or they can get it from LDL molecules. Okay, so I'm going to start with the absorption of LDL molecules via the LDL receptor. Okay, so we'll start off with what actually is an LDL molecule. Okay, so LDL stands for low density lipoprotein. Okay, and basically these are little spheres of lipids which can transport lipid molecules around the bloodstream. You see, there's a problem with transporting lipid molecules around the bloodstream, which is that lipid molecules are extremely neutral and therefore are extremely hydrophobic. They don't dissolve well in water. Okay, so what you do is you package them into these structures called lipoproteins, which then can interact nicely with water, and I will show you how they can interact nicely with water. Okay, right. So, basically low-density lipoproteins are quite small little spheres. Okay, they're reasonably small compared to some of the other lipoproteins, such as very low-density lipoproteins and chylomicrons. Their diameter is generally between 20 and 25 nanometers. Okay, and their structure basically consists of an outer layer. Okay, so the surface of the lipoprotein is made out of an outer layer of phospholipids. Okay, and generally phospholipids are drawn like so. They have a head which is polar and is usually drawn as a circle. Okay, and then they have these two hydrophobic tails which then are going to dangle into the center of the lipoprotein molecule. So the head is polar and will interact nicely with water, and therefore the entire surface of the lipoprotein is going to have these polar heads of the phospholipid molecules facing out towards the water, whilst the hydrophobic tails of all of these phospholipid molecules face into this center, where we're going to have a store, basically, of hydrophobic molecules. Okay, so let me discuss the main example of a phospholipid which is used within this phospholipid monolayer. Okay, so this outer layer consisting of a single layer of phospholipids is called the phospholipid monolayer. Okay, so the main phospholipid that is used in the phospholipid monolayer is a phospholipid called lecithin, or also called phosphatidylcholine, so I want to discuss that with you now. But before we just do that, let me tell you about what the actual definition of a phospholipid is. So what characteristics do you need to have to be a phospholipid? What are the criteria? Well, basically, one of the criterion is that you need a phosphate group within your structure. Okay? Generally, this phosphate group will be within the polar head of the lipid molecule. Okay? Because phosphate groups have a negative charge on them and therefore are extremely polar and interact nicely with water. Okay? So you need a phosphate group. And the only other criterion is that within your structure you also need some long-chain carboxylic acid, or a fatty acid, basically. Okay, so that's pretty lenient conditions. Many different molecules satisfy those conditions. Okay, one of the major groups of molecules which do satisfy the condition of being a phospholipid is the group of phosphoglycerolipids, of which lecithin, this main one that will be within the phospholipid monolayer of low-density lipoprotein, is an example. So we're going to firstly discuss what phosphoglycerolipids are so that we can see what the structure of lecithin is. Okay, so basically, phosphoglycerolipids are all based on a molecule called phosphatidic acid. Okay, so phosphatidic acid is the first structure we want to discuss, and phosphatidic acid is an example of a phosphoglycerolipid, and all the other phosphoglycerolipids are derivatives of phosphatidic acid. Okay, so let me show you the structure of phosphatidic acid. So basically what you have is two long-chain carboxylic acids esterified to the first and second alcohol groups of a glycerol molecule. Okay, so in green here, this vertical line that I'm now colouring in green, this is a glycerol molecule. That's what that vertical green line is representing. Okay, and the the organic chemist name for a glycerol molecule is to call it a propane 1,2,3 triol molecule. Okay, so glycerol is the old biochemist name for a molecule that is more correctly called propane 1,2,3 triol. Okay, and of the, firstly let's discuss the structure of this molecule. So the reason propane 1,2,3 triol is a better name for this molecule is it tells you exactly what the structure of it is. It tells us that we're dealing with a 3-carbon molecule where you have alcohol groups coming off the first, the second, and the third carbons. Okay, so each carbon of this 3-carbon molecule has an alcohol group coming off it. And then, to create phosphatidic acid, what you do is you take long-chain carboxylic acids, so carboxylic acid molecules which have got an extremely long tail, basically, and you esterify these to the first and the second alcohol groups of the glycerol molecule. So the old biochemist name for long-chain carboxylic acids is just to call them fatty acids. Okay, but again, long-chain carboxylic acids, which is the more correct name, is a more useful name because it tells us exactly what we're dealing with here. It tells us that we're just dealing with a carboxylic acid molecule which has an extremely long tail, basically. So let me give you an example of a fatty acid, a long-chain carboxylic acid. So the sort of archetypal example of a long-chain carboxylic acid would be stearic acid. Okay, and again, stearic acid is the old biochemist name for a molecule that's now more correctly called octadecanoic acid. And again, octadecanoic acid is a very useful name because it tells us exactly what we're dealing with here. It tells us that we're dealing with an 18-carbon, fully saturated carboxylic acid. So here is the carboxylic acid group. Now we need 17 more carbons, so 16 of these are going to be in the form of methylene groups within the middle. Okay, so here's a methylene group, and because I don't have enough space to draw 16 methylene groups, what I'm going to do is bracket this single methylene group here and subscript it 16 to say repeat this methylene group 16 times, basically. And then right on the end you have the final 18th carbon, which will be in the form of a methyl group, like so. So this is the structure of octadecanoic acid or stearic acid, and hopefully you can appreciate that it's a carboxylic acid molecule with an extremely long tail of 17 carbons, and that tail is extremely hydrophobic, it's extremely neutral because carbon and hydrogen atoms have pretty much the same electronegativity, so the electrons in these bonds are not sitting more towards the carbon versus the hydrogen, or the hydrogen compared to the carbon. They're sitting pretty much in between, so it means that the whole structure is nicely neutral, which is exactly the opposite of water, and means that water would much rather interact with other water molecules rather than the hydrophobic chain of this octadecanoic acid. So, what you're going to do is take two long-chain carboxylic acids, and they don't need to be the same long-chain carboxylic acids, they can be different long-chain carboxylic acids, okay, and you're going to esterify them onto the alcohol groups of the first and second carbon of the glycerol molecule. So let me just show you the esterification reaction. So, here is a carboxylic acid molecule, and now I'll just put an R there for arbitrary chain there, basically. and then we have our alcohol group of the glycerol molecule here. And basically, to form an ester link, what you do is you take the alcohol group of the carboxylic acid group, you take the hydrogen off the alcohol group, you bind those two things together to make water. Okay, so this is a condensation reaction because one of the products is water. And then you bind this carbon of the carboxylic acid group to the oxygen of the alcohol group to create what's known as an ester link. Okay, so you take two different long-chain carboxylic acids and you esterify them onto the alcohol groups coming off the first and the second carbons of the glycerol molecule, and that will create you what's a molecule that's known as a diacyl glycerol. Okay, so we're not quite at phosphatidic acid yet. What we've drawn so far is just a diacyl glycerol, sometimes just called a diglyceride. Okay, now why is it called a diacyl glycerol? What does acyl mean? Well, basically, if you notice, we have not added the full carboxylic acid molecule onto the alcohol group when we perform this ester link. The portion of the carboxylic acid molecule that we've actually added is this portion that I'm now circling in green. Okay, we have left off this alcohol group of the carboxylic acid molecule, okay, and only added the portion in green, and this portion in green is called an acyl group of the carboxylic acid. So it's a general term for any sort of group like this. It doesn't matter what the R group is there, it's called an acyl group basically. So we have actually attached two acyl groups onto glycerol, hence di for two, acyl for the acyl groups, and then glycerol. And diacyl glycerols are often abbreviated to DAG for short. Okay, right. Now, back to phosphoglycerolipids and phosphatidic acid. We haven't even got to phosphoglycerolipids yet. We're doing phosphatidic acid. Okay, so now, to turn a diacyl glycerol molecule into a phosphatidic acid molecule, what you need to do is add something onto the alcohol group that comes off this third carbon of the glycerol molecule. And basically what you're going to add is a phosphate group. Okay, so this little circle that I'm now colouring in purple here, this represents a phosphate group. Okay, so this is why phosphatidic acid can be considered a phospholipid, because it satisfies those two criterion that I wrote up earlier. It has a long-chain carboxylic acid within its structure and also a phosphate group. So, how to attach phosphate groups to alcohol groups? Well, let me show you the structure of a phosphate group. So basically, the structure of an inorganic phosphate group looks like this. You've got a phosphorus atom at the centre, double bound to an oxygen atom, with two alcohol groups coming off the sides, and then a single bond to another oxygen atom, which has acquired an electron via ionic means and therefore has a negative charge. Okay, then we've got the alcohol group that comes off the third carbon of the glycerol molecule. Okay, so basically we are going to bind this phosphate group onto this alcohol group of the third carbon of the glycerol molecule. Now, how do we do this? Well, if you look at this structure that I'm about to highlight in blue here, okay, so the phosphorus atom with an oxygen double bound to it and an alcohol group, what does that look like? That looks very much so like a carboxylic acid group. In fact, if I replace that phosphorus atom with a carbon atom, it would be a carboxylic acid group. Ignore the fact that it's got 5 bonds coming off it, which is clearly absurd for carbon. But if we just look at the portion that I've highlighted in blue, that would look exactly like a carboxylic acid group. So what can actually happen is these phosphate groups can interact with alcohol groups in pretty much identical ways to the way that a carboxylic acid group would interact with an alcohol group. So what you can do is you can break that alcohol group off the phosphate group. You can break the hydrogen off the alcohol group. You combine those two things together to make water. Okay, so again it's going to be a condensation reaction. And then you combine this phosphorus atom of the phosphate group onto the oxygen of the alcohol group. And that's very similar to an ester link. Okay, so this sort of a link would be called a phosphoester link. So this is a phosphoester link to parallel it with an ester link. Okay, right, so that's how we can add this phosphate group onto this alcohol group of the glycerol molecule, basically. And that now creates us a molecule called phosphatidic acid. Now, you will notice that there is not just one phosphatidic acid molecule, because we didn't specify what long-chain carboxylic acids we wanted esterified to our first carbon's alcohol group and to our second carbon's alcohol group. So there is scope for variation there, basically. You can put different long-chain carboxylic acids in these two positions, and it will still be called a phosphatidic acid molecule. So phosphatidic acid is really a huge class of different molecules that all have similar chemical properties, because, you know, the length of this chain doesn't really affect what this molecule is going to do. Okay, right. So the other thing I want to just discuss before going on to the more general phosphoglycerolipids is what's the difference between phosphatidic acid and phosphatidate. Okay, so basically phosphatidate is the conjugate base of phosphatidic acid. Okay, and let me explain what that means. So if we look at our phosphatidic acid structure, we have got this alcohol group coming off the phosphate group of the phosphatidic acid molecule. Okay, this alcohol group can lose its proton. Okay, so the proton on this oxygen can fall off the oxygen and go into solution, leaving the oxygen with a negative charge. Okay, so that's why phosphatidic acid is considered an acid, because the definition of an acid molecule is that it is one which can donate a proton into the solution. Okay, now, once the phosphatidic acid has donated its proton into the solution, what you have left is most definitely not an acid molecule anymore, because if you think about it, that oxygen now has a negative charge on it. Okay, so where is this molecule going to lose a proton from? It can't anymore. It can't donate protons into the solutions. It's certainly not an acid molecule anymore. The deprotonated form of phosphatidic acid is not an acid. It's actually a base, because this oxygen, which has a negative charge, can accept a proton out of solution again. So we call it the conjugate base of phosphatidic acid, because we got it by deprotonating phosphatidic acid, and the phosphatidic acid, once it's given away its proton, is then a base, and we call it the conjugate base of the phosphatidic acid. Okay, so this is a running theme for all acids, that once they've donated their proton away, what's left actually is a base. Okay, and the molecule that you have left over once you have donated the proton away, which has a negative charge on this oxygen, is then referred to as phosphatidate. So phosphatidate means the conjugate base of phosphatidic acid. Right. Okay, so in solution, okay, usually under physiological pH, phosphatidic acid does donate its proton away, so usually in the cell what you will find is that most of the phosphatidic acid molecules are actually present in the conjugate base form, so they're present as phosphatidate. So people will often talk about phosphatidate rather than phosphatidic acid. Okay, right. So, that's phosphatidic acid and phosphatidate. Let's now talk about what a phosphoglycerolipid is, and then we'll look at specifically our main example, which is lecithin, which makes up this phospholipid monolayer of lipoproteins, but specifically the low-density lipoprotein, which we're interested in. So basically, to turn phosphatidic acid into a more general phosphoglycerolipid, what you need to notice is that if I now circle this portion in turquoise here, this portion will be still intact once you have linked this phosphate group via a phosphoester link to this alcohol group on the third carbon of the glycerol molecule. Okay, so this turquoise portion is still intact. And what does that look like? It looks like one of these carboxylic acid-like groups. Okay, so in fact this can now react with an alcohol group again. So phosphate groups can react with alcohol groups to form phosphoester links twice, because they effectively have two of these carboxylic acid-like groups within them. Okay, so they're better than carboxylic acids at doing this. They can do it twice. Okay, so if we bring along another alcohol group here, what we can do is we can take that alcohol group off the phosphate group. We can take a hydrogen off the alcohol group. We can combine those two together to make water, and then we can link that phosphorus atom to this oxygen atom down here to create another phosphoester link with another alcohol group. So basically, I can attach alcohol groups onto my phosphate group here, again via phosphoester links. Okay, and that's how you create a more general phosphoglycerolipid. You can attach alcohol group containing molecules onto the other side of the phosphate group, basically. So we now want to discuss the specific example, which is lecithin, which is also called phosphatidylcholine. And basically, I should say that whenever you make a phosphoglycerolipid like this, so you start with a phosphatidate or a phosphatidic acid molecule, and you esterify via a phosphoester link onto that phosphate group, a group containing an alcohol group. Okay, the molecule that you end up with, it has within its name the prefix phosphatidyl because it contains the phosphatidic acid molecule, basically. So phosphatidyl means that you've got all of this bit, and then you need to tell me what the additional group is. So we're doing phosphatidylcholine, which means that we are effectively taking a phosphatidic acid molecule and esterifying via a phosphoester link a choline molecule onto the other side of that phosphate group. Okay, so all I now need to tell you is what is the structure of choline. Well, we know one of the things it must have in it, which is an alcohol group. Okay, so let's just see the rest of its structure. So this is the same choline as is within the famous neurotransmitter acetylcholine. Okay, so in acetylcholine, what you have done is you've esterified the choline to an acetic acid molecule to produce acetylcholine, which is an ester. We just want the choline now without the acetic acid group added on. So here's the alcohol group. Then you've got an ethylene group, so just two methylene groups stuck onto one another. Okay, and then on the other side, you've then got a nitrogen atom, and this nitrogen atom has 3 methyl groups coming off it. 1, 2, and then a third one here. Now that leaves this nitrogen at the center here with 4 bonds coming off it. Okay, nitrogen doesn't like having 4 bonds. Okay, it likes having 3 bonds. So into one of these bonds, nitrogen has basically donated both electrons. So the normal understanding of a covalent bond is that 1 electron comes from the nitrogen and 1 will come from the carbon. So 1 electron from each of the 2 members of the bond. Okay, however, in one of these bonds, let's say it's this one here between this nitrogen and this carbon, the nitrogen has donated both electrons. So it's donated its lone pair into this bond, basically. Okay, now, what does that mean? Well, as I say, the usual understanding of a covalent bond is that 1 electron belongs to the nitrogen and 1 belongs to the carbon. So if the nitrogen puts both in here, it's as though the nitrogen has given an electron away to the carbon. Okay, so that's going to leave the nitrogen with a positive charge, basically. And you might ask, well, does that mean the carbon's going to have a negative charge? Because, you know, it's received the electron from the nitrogen. And the answer to that is no. And the reason is that when this carbon originally came into this sorted arrangement, it will have had a positive charge, so it will have come in with a positive charge. It will have then received the electron from the nitrogen when it formed this covalent bond. It will have then had its positive charge neutralized, and it's effectively deferred the positive charge onto the nitrogen atom. Okay, so choline overall then has a positive charge. Okay, so this is the structure of a choline molecule. So all that you need to do to create phosphatidylcholine is esterify this alcohol group onto the other side of the phosphate group of a phosphatidic acid molecule, and then you've got phosphatidylcholine. Okay, now, this is a phospholipid, okay, because we've got a fatty acid and a phosphate group. Now, it's a perfect example of a phospholipid because the phosphate group has this negative charge here, so that's very polar, okay? Then this choline group that we've added on here, this also has a positive charge, so that's very polar as well. Whereas this portion down here, where we've got the 2 long-chain carboxylic acids linked down here, these are very hydrophobic, so those represent the hydrophobic tails. So this molecule really can be represented like so. This is the polar head with the phosphate group and the choline. These are the 2 long-chain carboxylic acids, which are extremely hydrophobic. So, phosphatidylcholine makes up most of the phospholipid monolayer that surrounds low-density lipoproteins. So you have a layer of these phosphatidylcholine molecules. Okay, right, so we'll call it there for this video, and we'll continue our discussion of the SREBP pathway in the next video.